video, I'm going to be talking about the process of development. So we've talked about how fertilization occurs and how essentially you have two haploid organisms coming together to form a diploid organism. Here we're going to give some of the insight into the mystery of what happens as a single cell organism essentially is going to become a multi-cellular organism with very complex structures with cells that are very different from each other, even though they contain the same genetic information. So development is a process that begins upon fertilization, but will continue until after the individual is born, and can really be seen as a continuation all the way up until death, because the organism, in this case the human being, is constantly going to be changing throughout their lifetimes. So we're going to talk about, especially focusing on the beginning of this, what is happening after fertilization occurs. So the after fertilization occurs, the zygote and its membranes are together called the conceptus, and it starts to move towards the uterus. This is going to be driven by a couple of things. First of all, you have peristalsis that is occurring within the uterine tube, and you also have ciliary beating of the epithelial cells that are lining the tube. Now, as it is moving towards the uterus, it's going to start dividing. So these divisions are known as cleavage divisions. Cleavage divisions are a rapid mitotic division that occurs that increase the cell number but doesn't overall increase the size of the conceptus, because they are not growing in between these stages. So you go from a zygote to the 2-cell stage, then the 4-cell stage, moving on to the 8-cell stage, the morula, and then finally you're going to have after that a more growth-related phase. So each division of these is going to result in a cell that is known as a blastomere. The morula is going to reach that stage of about 16 cells about 3 days after fertilization. It's called a morula because that means little mulberry. If you've ever seen a mulberry, it kind of looks like a raspberry with just a bunch of circles all bumping into each other together. That's what the morula looks like. So the morula is going to float freely in the uterus for several days, and then will continue dividing while consuming uterine milk. Uterine milk is going to be secretions that come from the endometrial cells that can be used for a nutritious source of energy and resources for growing. Once after it's reached this, the conceptus is going to keep dividing until it reaches about 100 cells, at which point it's going to organize itself into what is known as a blastocyst. So this is going to be consist of cells that are known as trophoblast cells surrounding them, making a circle. In the middle of the circle, you have a fluid-filled cavity that's known as the blastocele, and you have some cells that are lining the inside of it, known as the inner cell mass. The inner cell mass is what is going to become the embryo, while the trophoblast is going to form the chorionic sac and contributes to the fetal portion of the placenta. At this point, the inner cell, the cells of the inner cell mass are totipotent, meaning that they can differentiate into any cell type, but that's only going to last for a few days. They're going to start to become pluripotent multipotent and eventually unipotent cells as they differentiate, which means that when they divide, they're going to be limited in what they can actually become as far as skin cells or a liver cell or a neuron and so forth. By the end of the first week, the blastocyst contacts and adheres to the uterine wall. It does have to hatch and shed its zona pellucida by this. It will rotate and implant into the endometrium of the uterus. This typically occurs in the fundus of the uterus, but can also occur in the posterior uterine wall. Implantation may cause some minor bleeding. There are a few issues that can arise in this. So if you have the blastocyst implanting elsewhere, it can result in an ectopic pregnancy. This is going to be a pregnancy that is not ultimately viable for the developing organism. And will oftentimes result in a spontaneous miscarriage, but sometimes surgical removal of the organism is necessary in order to save the mother's life. The blastocyst fails to implant anywhere from 50 to 75% of the time, meaning that really at least half of the blastocysts are going to be shed during menses. This is why it may actually take multiple ovulation cycles to actually achieve pregnancy. Upon implantation, the superficial trophoblast cells are going to fuse into what is known as a syncytial trophoblast. So this is a multinucleated layer that digests the endometrial tissue and anchors the blastocyst securely to the uterine wall. At the same time, the uterine mucosa is going to grow around and envelop the blastocyst. So HCG secretion is going to begin shortly after this. So the trophoblast is going to secrete human chorionic gonadotropin, which is going to help maintain the corpus luteum. So let's zoom in and look at what's going on during this time. So the blastocyst digests the uterine mucosa. Eventually, the endometrium is going to grow around the embryo, fully securing it. The implanted embryo continues to grow within the endometrium after conception. And as mentioned before, the most common site of implantation is going to be the fundus or the posterior uterine wall. All right, the trophoblast is going to be made up of two different types. So you have the cytotrophoblast. These are the individual cells. The syncytial trophoblast are going to be the multinucleated cells that make that up. So the trophoblast cells here are going to secrete the corpus luteum, or secrete the HCG, which helps maintain the corpus luteum. HCG is also going to prevent menstruation and stimulate continued secretion of progesterone and estrogen. HCG is the hormone that is the basis for the pregnancy test. So by the time implantation has occurred, that is typically around the middle of the second week, and at this point enough HCG will be present for a positive test within just a few days after implantation has occurred. All right. So during the second week of development, the implanted blastocyst cells differentiate into layers. Some cells are going to form extra embryonic membranes that support and protect the embryo. So you're going to have things like the amnion, the yolk sac, the allantois, and the chorion. The inner cell mass is going to differentiate into the hypoblast and the epiblast. The hypoblast is actually considered the lower layer, and the epiblast is considered the upper layer, so this is closer to them. A space that is going to develop between the epiblast and the trophoblast is going to be what becomes the amniotic cavity. So epiblast, this is going to be considered the primitive ectoderm. This is the source of all three definitive germ layers, which are the ectoderm, the mesoderm, and the endoderm that arise during gastrulation. The hypoblast is considered the primitive endoderm. So it's positioned ventrally towards the blastocyst cavity and mainly forms extra embryonic tissues like the yolk sac, thus playing a key role in setting up the embryonic body plan, including inhibiting premature primitive streak formation. I mentioned gastrulation. That is a process that occurs during week 3 when epiblast cells are going to migrate through what is known as the primitive streak to replace the hypoblast and fill the middle layer, forming what is known as the mesoderm. All right, so as I was mentioning some of these structures before, we have the amnion. This is formed from the epiblast cells that surround the amniotic cavity. It becomes a membranous sac by the end of week 2 and fills with amniotic fluid, eventually surrounding the embryo. Amniotic fluid is an early filtrate of maternal plasma, but after about week 8, fetal kidneys are going to start adding urine. The function of the amnion is to cushion the embryo and the fetus from trauma. It protects from temperature changes that may occur, allows for free movement, and facilitates practice of the processes of swallowing, breathing during development. The yolk sac forms on the ventral side of the embryonic disc. It is going to develop from hypoblast cells extending into the blastocyst cavity. It will provide nutrients absorbed from the trophoblast and supply primitive blood circulations during the during weeks 2 to 3. By week 4, the placenta is actually going to take over nourishment, and the yolk sac will start to shrink. It does have later functions as a source of early blood cells and a source of germ cells, which are your gamete-forming cells, the oogonia and the spermagonia that gave rise to the egg and sperm. The allantois develops during week 3 as a finger-like outpocketing of the yolk sac. It is going to serve as a primitive excretory duct that contributes to and ultimately contributes to the formation of the urinary bladder. Along with the yolk sac, stalk is going to help form the outer structure of the umbilical cord. And then the chorion is the outermost embryonic membrane and encloses all of the other layers. It plays a major role in placental development, and further development of the chorion is tied to the formation of the placenta. As I mentioned, gastrulation is going to be the process that gives us the 3 primary germ layers: the ectoderm, the mesoderm, and the endoderm. These layers will ultimately give rise to all tissues and organs of the body. So the endoderm is going to be the first layer formed. It replaces the hypoblast and lies adjacent to the yolk sac. The mesoderm is going to form the middle layer and occupies the space between the endoderm and the ectoderm. And the ectoderm is going to be made up of epiblast cells that ultimately do not migrate and remain on the embryo surface. So the endoderm layer, these are going to become a large number of your visceral organs, such as those that make up your digestive system, the liver, pancreas, and lungs. The mesoderm, this is what ultimately is going to give rise to a large portion of your muscular and skeletal systems, as well as the circulatory systems. And the ectoderm is going to become things like your hair, nails, and skin, and most importantly, your nervous system. So your brain and spinal cord are going to be derived from the ectoderm layer. Here we can see a big picture summary of what's going on. So the early embryo is going to be nourished by maternal endometrial tissue. From weeks 4 to 12, the placenta will gradually develop. A mature placenta is going to have both a maternal component, which is known as the decidua basalis, and a fetal component, which is made up of the chorionic villi. The chorionic villi are the structural and functional core of the placental exchange, while the umbilical cord is going to serve as a circulatory link between the fetus and the placenta. Early nourishment is going to happen by decidual cells, so this is weeks 1 to 4. After implantation, the embryo is initially nourished by these cells that are specialized cells of the endometrium. You don't have a functional placenta yet, and you need secretions from the maternal tissue as well as diffusion. You have a transitional period between weeks 4 and 12, where the placenta is gradually going to assume the role of nourishing the embryo, but the decidua cells are still there. They're going to slowly become less and less needed for nourishment as the embryo transitions to a blood-mediated exchange system with the mother. And then the mature placenta is derived from both the fetus and the mother. As I mentioned, the maternal portion is going to be derived from the decidua basalis, which comes from the endometrium, while the fetal portion is derived from the chorion, which is derived from the trophoblast and to some degree the mesoderm. The umbilical cord structure is going to connect the conceptus to the placenta. 2 umbilical arteries are going to be involved in this that are going to carry deoxygenated blood and waste flow from the fetus to the placenta, and 1 umbilical vein that carries oxygenated blood and nutrients where the flow goes from the placenta to the fetus. You have other supporting structures such as the amnion that surrounds the umbilical cord, Wharton's jelly, which is a mucus connective tissue that is going to protect blood vessels from compression. The embryonic portion of the placenta is going to form from the syncytial trophoblast. That's the outer multinucleated trophoblast layer that invades the maternal issue, and the cytotrophoblast, which is the intercellular trophoblast layer that actively proliferates along with the extra embryonic mesoderm. All right, so as the maternal blood bathes the chorionic villi with their intervillous spaces, the fetal blood is going to flow through vessels inside the villi. There's no direct mixing of maternal and fetal blood. Instead, exchange is going to occur by diffusion, active transport, and facilitated transport. So if we look at this, we can see the placenta is delivering nutrients to the fetus and taking waste away from it. First of all, there are a number of different structures that are going to aid this process. So blood arriving via the umbilical vein, going to be oxygen rich. Notice that we have an opening that's known as the ductus venosa. So the oxygenated blood from the placenta will go away from the semi-functional liver and instead move towards the heart. So that is the first shunt that is ultimately carrying blood to the heart via the inferior vena cava. The oxygenated blood from the placenta is going to enter in via the right atrium. So we can see here, you're going to have mixing of the blood that's coming in from the superior vena cava. Ultimately, you have the foramen ovale, which is going to allow oxygenated blood in the right atrium to reach the left atrium. And you have blood flow thus to a degree circumventing the pulmonary circulation. So it goes to the right atrium, into the left atrium, then into the left ventricle, ultimately going up through the aorta. As it goes through the aorta, the ductus arteriosa, which connects the aorta with the pulmonary artery, is going to further shunt blood away from the lungs and go into the circulation. Thus, mixed blood travels to the head and body and then back to the placenta via the aorta. So, as mentioned, you have a few of these shunts occurring before birth. So the foramen ovale and the ductus arteriosus and the ductus venosus are all present before birth. After birth, the ductus arteriosus is going to constrict, allowing all blood leaving the right ventricle to travel to the lungs via the pulmonary arteries. Thus, the pulmonary and also the foramen ovale is going to close, leaving a small depression known as the fossa ovalis. The ductus arteriosus is going to become the arteriosus ligamentum. So it's sort of like a ligament that is forming between those two structures that remains there. The fossa ovalis that forms here is going to prevent deoxygenated and oxygenated blood mixing within the heart. And then the ductus venosa is going to degenerate and become the ligamentum venosum. The inferior vena cava is now only going to carry deoxygenated blood back to the heart. All right, so that was an overview of the process development, ending with how the fetus handles circulation and how that changes after birth. So that's the end of this video, and I will see you guys next time.